Fungi definition, no need. Fungi definition. What you know about fungi? Tell me that much enough.
It is used in like bread.
It is
Used in yeast, for example.
I'm asking about general definition.
It is eukaryotic,
Okay, eukaryotic, right?
non-chlorophylls,
Eukaryotic, okay.
and they are saprophytes. Well.
Are you able to see the slide?
No, sir.
Just wait. Now clear?
Yes.
Now tell me the fungi definition. Have you seen ever in our daily life fungal members?
Yes, sir.
Where did you see? So we can see on rotten vegetable and fruit mainly fruit and also well-known one mushrooms. You can see in a slide PPT slide also. They are all belongs to fungi, right? The fungi, as you said, these are eukaryotic and these are autotrophs or heterotrophs. Fungi are auto or
Heterotroph.
heterotrophs.
Heterotroph.
Heterotrophs, lack of chlorophyll, achlorophyllus, lack of chlorophyll, they are heterotrophic. So the simple definition, the fungi are achlorophyllus or heterotrophic eukaryotic thallophytes. The body of the thallus. What are thallophytes? I already told you in
Yes.
algae. What is thallophytes?
Does not have any structure.
Thallus-like structure means there no proper structure, like there is no proper root and shoot and leaf. They
Yes.
are all together. That type of called as a thallus, right? So in this topic, in general character, first we follow the order. Already told you the order. What is the order? First heading, where they are,
Habitat.
how they are. Second how they are, and second how, and third aspect is how can they reproduce. So these three important. The first where they are means occurrence or habitat. The natural occurrence or habitat. So generally habitat or occurrence. First one. Next, how they are means thala structure. In the after thala structure, what we learn in order. Don't forget the order of headings.
After
Habitat
that is
occurrence,
cell
thala,
organization,
right? Cell structure. After cell structure,
reproduction.
reproduction. That much enough. Right now, they are the fungi are cosmopolitan one, which means they can grow everywhere,
Everywhere.
everywhere. Maybe in air or water and soil, in plants and animals, so on. So mainly they are preferred to grow in warm and humid places. Hence, we keep food in the refrigerator to prevent the bacterial and fungal infection, right? Because there is no warm and humidity in the inside the refrigerator. That's why the eradicate these fungal members, especially and also the bacteria members. We generally store the food material inside the refrigerator, right? After the occurrence, habitat, the next one is thallus organization. If you see the thallus, here we can see in diagram, this is also the fungal member, this type of also fungal member. And they can grow on the plant. Now let's see the thallus structure. Maybe this is unicell form. Here the formation like a algal member, unicell to multicell. Unicell to multicell. You need to multi-cell. Remember it. You need to unicell example yeast. Best example yeast. And the rest of the all multicellular. And here the fungal body is thallus and a name given as a mycelium. Mycelium, the vegetative body called as mycelium. That means thallus structure called as mycelium. Let's see the mycelium structure. Mycelium is an interwoven mass thread like this. Mass thread like hyphae, singular hypha. That means actually this is one part. For example, actually here not given. Uh, do you see this diagram? Blue color diagram. What you drawing side? Hello,
Yes, sir.
Yes, sir.
right. For example, this is the fungal mycelium. Septa present. Maybe septate or aseptate. Okay, that is based on the class and species here. So now, and also can draw another one also like this. So one, this one cell, it is called as IFA. This is called IFA. This is called IFA. And this total group called IFA. One, two, three, four, called IFA. And the entire body called as mycelium. Entire body called as mycelium. Is it clear? Vegetative structure. That means the basic unit, base like a simple. Uh, Actually, are we made up of? Generally, organisms made up of by. Hello.
Yes, sir.
What is the basic unit of the an organism for structural
Cell.
cell? Like a cell here, given name as a hypha. That is
Yes.
singular. And this group of hypha and form as a hyphae group. And all hyphae together form as a thread-like appearance. This thread-like appearance called as mycelium, right? So vegetative body mycelium, which is made up by hypha. That's it. These hyphae may be septate. I just now told you may be septate with cross wall, like this cross wall. Here in diagram there is no septa. That is called aseptate, right? Maybe septate or aseptate. So that is the based on the species, right? Is it clear? Vegetative structure clear or not?
Yes, sir. Yes.
Right. Now after vegetative structure, what we learn? Filamentous are again given the unicellular, holocarpic, and just wait. So in unicellular, the vegetative body that is called as holocarpic. Here you can remember the two words such as eucarpic and holocarpic. Just wait, right. So here given eucarpic, sorry, holocarpic, and where the given eucarpic. Okay, remember it, eucarpic and it holocarpic. Simple. Holocarpic in general, what happened? Here the vegetative and reproductive part separated. If the vegetative, for example, this is vegetative body, on the vegetative body, reproductive, this round shape reproductive part is present. This type of structure called as eucarpic. But what happened in unicell? Unicell, the cell made up of a single, it's completely itself single one. It acts as a vegetative, it acts as a reproductive cell. The type of cell called as a holocarpic. So in the unicellular holocarp form, mycelium is absent. There is no mycelium. Only the example, best example, fungi. Fungi, the simply single cell. That's it. It's single cell. We confined as a vegetative and a reproductive structure also. So one cell both perform together. That means form the both structure. That is called holo. Both function. That is called holocarpic. Right? And filamentous thallus. Filamentous composed hyphae already discussed about this. And aseptate or septate and aseptate also we also completed. Next, right now the different forms of mycelium. You know, in our book also given the mycelium different forms. Let's see. Just wait. Are you able to see different forms of mycelium?
Yes, sir.
Hello? Right.
Yes.
Plectenchyma, one type of mycelium. Prosenchyma, one type mycelium. Pseudoparenchyma, sclerotia, and rhizomorph, rhizad, appressorium, haustorium. So stomata. These are the most different type of mycelium. First, with diagram, discuss about plectenchyma. Actually, in diagrammatically, plectenchyma not given, but anyway, right. So this type of mycelium, the fungal mycelium, if organized by loosely or compactly woven to form a tissue called plectenchyma. This plectenchyma, two types. So how is arrangement of mycelium organized loosely or compactly? It can arrange loosely or compactly. So in this two type, prosenchyma and pseudoparenchyma. Here the prosenchyma cells, we can see in diagrammatical prosenchyma, the fungal tissue hyphae loosely interwoven, more or less parallel to each other. So this is loosely interwoven and they are arranged in parallel. This type of arrangement called as parallel, and they are not a firmly attached, they are loosely attached. And pseudoparenchyma, if you see pseudoparenchyma, just right. Pseudoparenchyma is this fungal tissue hyphae are compactly interwoven. Compactly, compactly, you can see in diagram. If you see here the arrangement of the compactly interwoven. Just wait. Right. So here first loosely interwoven prosenchyma, same compactly interwoven, that is paraplectenchyma and pseudoparenchyma. This is not loosely arranged. This is compactly arranged, and they are not a parallel. In the first diagram, parallel arrangement, that is prosenchyma. And round shaped cell look like a parenchyma tissue. It is look like a parenchyma tissue. In section cutting, it look like appeared as a parenchyma tissue, but not true. That's why it is called pseudoparenchyma. And another one, sclerotia. We can see here in diagram the black color structure called sclerotia, like a thread-like structure. If you see the sclerotia, the mycelium hard, mycelium very hard, dormant bodies, and very compact and protected by external thickened hyphae. Like a rope, remember, like a hard rope. That means the entire mycelium stands firmly attached like a thread, like a thread. And they sustain during unfavorable condition. When the mycelium return favorable condition, that means when the favorable condition reoccur, then grow as a mycelium. That is sclerotia. So simple that is simple only the mycelium arrangement. Mycelium arrangement. If the mycelium arrangement parallel and loosely arrangement, that is prosenchyma. Compactly arranged and round in shape, that is called pseudoparenchyma. And the sclerotia very firmly attached. That means one like a threads. We interwoven the threads to make a rope. Different type of threads compactly arranged to make a rope, right? Like this here also the mycelium threads, mycelium hyphae, they are interwoven, hardly interwoven and form a like a rope-like structure. It is appear as a hard body, external thickened hyphae, That type of structure called as sclerotia. We can see in diagram also. This is called sclerotia. And next one, rhizomorph. Rhizomorph, root-like, compactly interwoven. Root-like, root-like, compactly interwoven. You can see in diagram also here. So somewhat strong than rhizomorph. Sorry, than sclerotia. Right? They are compactly interwoven hyphae with distinct growing tip. Distinct growing tip. Develop of absorption vernations, example armillaria. So this root-like appearance. So anyway, the threads, that mean fungal thread, fungal hyphae, all together form as a root-like appearance. So this is called rhizomorph. This structure called as rhizomorph. And actually rhizoids, not a rhizoids, one type of mycelium part. So in rhizoids are short root-like filamentous here. We can see it here. The short root-like filamentous branch thallus generally formed of base thallus also function as anchoring and also absorbing. That's it. Aprosorium. It is a swollen mucilage structure infected hyphae. Mainly it forms the host and by it, which is formed by the parasitic fungi, which is adhere to surface to the host or other substratum and helps the penetration to the into the host, penetrate into the host. Like this. You can see diagram. Right? Terminal. This is aprosorium. Outer. This is aprosorium. This is the host body. Host body. Host body. Simple lobed. This is called lobes. Swollen mucilaginous structure of infection hyphae. This is infection hyphae. Infection hyphae. Actually, infection hyphae present inside the host. From present inside the host, there are some external parts like a swollen balloon-like structure formed. This is called aprosorium. This aprosorium, which is adhered to the surface of the host. Surface, this is surface area, surface of the host. Surface of the host. Right, just wait 1 minute. And it helps in penetration of the infection hyphae. From this infection hyphae, penetrate into the host. What is the function of this infection hyphae? Function. From this infection hyphae, they can absorb the food material from the host. And another one is hostorium. Hostorium is an organ that is developed in same like aprosorium from hyphae, usually performing the functioning absorption same like aprosorium. But they are mainly present in obligate parasite. I will tell you later the fungi nutrition, what is obligative, what is facultative. They are vary in shape, maybe knob-like or button shape, elongated, branched. We can see here the branched. You can see in branched. Right? And they secrete some of the enzyme which hydrolyze the protein and carbohydrates of the host plant. And after hydrolyze, they can absorb. So this is fungal hyphae. Fungal hyphae. What happened from the fungal hyphae? Some of the lobes branched members are emerged. These branched structure called as hostorium. So remember it. It look like some of the infection thread. In aprosorium, hostorium different. In aprosorium, they are formed out surface of the host, but hostoria inside the host, inside the cell. They mainly formed in the inside the cell. Only small some portion penetrate into the host, but not completely. But here almost above 50% penetrate into the host cell. Now the snails traps no need. That is look like a Hostoria, and where it is. And last one is stomata. These are compact somatic structure, much like matrices and fractification are generally formed in them. Same, these also we can see in these are stomata, the somatic structure, but very much compact. They are very much compact and look like a matrices. It is appears like a matrices. So this is all about the fungal mycelium variations. Now, after the thala structure, what is next? What we completed next? After thala structure, hello? Hello?
Cell structure.
Cell structure. Let's see the cell
Cell
structure.
organization.
Right. 1 minute. Cell structure. So you know the cell is eukaryotic, right? Tell me the what type of cell organelle present in eukaryotic organism. First of all, in cell structure, let me complete. Let us complete the first and components. What are the components of the cell in order? Tell
Cell
me in
wall.
order. Cell wall, right? Next,
Yes, cytoplasm.
after cell wall,
Plasma membrane.
plasma membrane. Next,
Nucleus.
nucleus,
Cytoplasm. No.
cytoplasm. All cell organelles present. What are the cell organelles?
Mitochondria, Golgi complex.
Mitochondria, Golgi complex, endoplasmic reticulum, except the plastids, all cell organelles present in fungi. Remember it.
Yes, sir.
Plastids, chloroplast, absent because these are not a autotrophs. Heterotrophs, as it heterotrophs, plastids are absent in fungal member. And cell wall present. Here the cell wall. This is the characteristic shape of the cell and protect that is the fundamental function of the cell wall. Protect the cell from the osmotic lysis and also binding site for the some enzymes. And chemically, which is made up of chitin. Chemically, the cell wall made up of fungal cell wall made up of chitin. The formula of chitin C22H54N4O21. And after that, the cytoplasm hyphatic and which has containing Golgi complex, chitosomes. These are the new cell organelle chitosomes. And earlier, as you said, all type of cell organelles also present. We can see in diagram also. Here, we can see the diagram. Endoplasmic reticulum, nucleus, all type of they are present. And here the reserve food material is reserve food. What is the reserve food material in plant? Reserve food material in plant.
Starch.
Starch. But in glycogen and oils, reserve food material, glycogen and oils, right? So this is about the cell structure, and this is cell diagram, and we can see in all type of cell organelles in this diagram: endoplasmic reticulum, mitochondria, lipids, and chitosomes, lomosomes also. Only chitosomes and lomosomes present in fungal members only. Remember chitosomes and lomosomes, right? Next one, fungi nutrition. Fungi nutrition. If see fungi nutrition, as heterotrophs, they cannot prepare their own food material. For food material, they can depend on others in the form of saprophytes and parasites and symbiotic. These three important. First, let's complete that. That means the most of the fungal members saprophytes. So first of all, let's see the saprophytic fungi. What are the saprophytes? We can see in diagram also. What is saprophyte here? What is saprophytes?
Obtain food from dead and
Right, they
decaying
obtain
plants.
food. In nutrition means obtaining food,
Yes.
getting food. The getting food, they can prepare their own some organisms. Some of the organisms depend on others. Fungi depend on others. Here the other may be other plant, other host, or maybe death and decay particle. Here the food obtained from the dead and decaying organic matter. They secrete digested enzymes to outside, which digest the substratum and then absorb the nutrient. Because actually, here the any matter in solid, if the matter, that organic matter in solid, they can't absorb. The fungal member can't absorb. That's why actually when infect, first of all, release some of the enzymes. These enzymes, the solid one makes it to liquid. Now this liquid nutrients absorbed by the fungal members, right? Example, mucor, agaricus, rhizopus, and second one is parasitic. You know parasite. What is parasite? Parasite that mean obtaining here the obtaining food dead, but here the obtaining food from living organism. Living organism. They may be here given facultative or obligative. Facultative parasite, obligative parasite. First facultative parasite grow variety of tissue. Example, Ustulago, and obligative parasite, downy mildew. Let complete about definition facultative parasite. What is facultative parasite here? Facultative means this is temporary parasite. Remember it. Facultative. Actually, this facultative parasite actually not a parasite. In general, naturally, these type of fungal members grow as a saprophytes. Actually, they grow as a saprophyte and they collect the food material from the death and decay particle. But whenever host is available, immediately they can turn into parasitic nature. Is it clear? Hello?
Yes, sir.
Right. So that type of condition called as facultative. Example, Ustulago. And obligative parasite. Here another one obligative parasite. They can survive only parasite, but not saprophyte. For example, if the host is available, they perform as a parasite and take the food material from the host material. If there is no host, what happen? If there is no host, what happen? Can they perform as a autotroph saprophytes? Can they survive as a saprophyte? This type of parasite, obligative parasite, can they survive?
No.
Can they survive like saprophyte?
No, they cannot survive.
No, cannot survive. They would be die. So that type of parasite called as obligative parasite. The example is downy mildew. Example downy mildew. Right? And it's the same saprophyte also same two types. Obligative saprophytes and facultative saprophyte. Say what is facultative saprophyte? Hello?
Temporary.
A temporary, but actually facultative saprophyte grow in general grow as a parasite or saprophyte.
Saprophyte.
Facultative saprophyte, they are not a saprophyte. They are parasite. They generally grow as a parasite, but if there is no host to depend, there is no host. Immediately they turns into saprophyte, and they can take the material from death and decay. That means the facultative saprophyte can survive death and decay particle and can survive on living host. Remember it, okay? Is it clear?
Yes, sir.
Yes.
Right. And next one, I think here reproduction. So symbiotic nature. What is symbiotic nature? Symbiotic. Symbiosis means mutually get both are get
Mutual.
beneficial. The best example for symbiotic already yesterday I told you lichen. What are the lichens? What are the lichen? These
They
lichens are symbiotic nature. What are the partners in lichens? Yesterday we discussed
Yes, sir.
algae and fungi. Algae and fungi here, the association between the algae and fungi, right? So this is called lichen and also mycorrhizas. Mycorrhizas also, that means the mycorrhiza, the symbiotic association between soil fungi and the roots of the plant, roots of the most plant. Root means rhiza, myco means fungi. Study of fungi called as mycology, right? Hello?
Yes, sir.
So that's why called as mycorrhiza. This is this mycorrhiza may be endomycorrhiza, ectomycorrhiza. Ectomycorrhiza means outside the root. Endomycorrhiza means inside the root. So this is the all type of, this is the different type of nutrition in fungi. This is also important for short answer. And heterothallism, homothallism, we will discuss later about, sorry, one, let me complete in reproduction. So simple here, the heterothallism, homothallism. The black seed discovered the mating type of genetically distinct strains in mucor, sorry. A name given hetero means different. Heterothallism, hetero means different, so different mating types. Some fungi and algae homothallic, both same simple here. Don't get confusing this one type of reproduction in heterothallism and homothallism. So for example, here the pink color different stain, green color different stain. Here the these two different stain mating, difference to mating. That is called heterothallism. In homothallism means same type, same species. Actually, for example, this is the plus thallus, plus mycelium. This is the minus mycelium. If the mating between plus and minus different strains, that is called heterothallism. The plus mycelium again mating with plus mycelium, that is called homothallism. That's it. Plus mating with plus mycelium or minus mating with minus mycelium, that is homothallism. Different strain called as heterothallism. Right? Next one, reproduction. Vegetative reproduction, asexual reproduction, and sexual reproduction. Tell me, vegetative reproduction, how they perform? Takes place by three methods: vegetative, asexual, and sexual. Actually, right. Vegetative reproduction types is here the given vegetative reproduction types. Already, fragmentation, and also fission and budding, sclerotia. What is fragmentation? Hello? We have been discussing since the algal class about fragmentation. Tell me what is fragmentation? There is less time. Fragmentation. Here, are you able to see the slide? Hello? Hello?
Yes, sir. The fragmentation, small pieces, it breaks and then grows in the suitable condition, germinates.
Right. The hyphae break into small space, fragment. Now each fragment can grow as a new mycelium. Right? That is called fragmentation process. And fission method involves splitting of cells into two daughter cells. We can see here.
Splitting of cells, yes, yes.
Right. Here the one cell splitting into two cells. That is called fission. And budding. Here the budding, the first, for example, this is the yeast cell. In yeast, what happens? Small bulb-like structure develop. Like this is called bud. And lateral, this bud structure
Separated.
gradually increased. Later it is separated from the mother cell. And this bud can develop into
Yes.
another yeast cell. And sclerotia type, these are pernating bodies, and formed by compact masses interwoven of hyphae. Just now we have discussed about types of mycelium. about the sclerotia. In sclerotia, interwoven hyphae, just wait, small call is there. Okay. Interwoven hyphae, the formed the, and these are under suitable conditions. Actually, this sclerotia, like a thread-like, strong appearance, formed during unfavorable condition. During unfavorable condition. But when favorable condition reoccurred, again, these sclerotia degenerated into mycelium. Right?
Yes.
Next one.
Asexual reproduction.
You have to. Rhizomorphs, rhizomorph also one of the reproductive part. These are root-like elongated mycelial strand. They remain dormant under the unfavorable condition like a sclerotia. When the favorable condition reoccur and develop into new mycelium. So sclerotia and rhizomorph both are formed under unfavorable condition. When the favorable condition reoccur, they can grow as a new individual thallus. That's it. And uh, these are all belongs to the vegetative reproduction. Now let's see the asexual reproduction. What is asexual reproduction? What is asexual reproduction? How the asexual reproduction takes place?
Through spores.
That's it. Asexual reproduction by means of spores. Simple. By through spores. Maybe the spores are motile or non-motile. Or conidia also. Conidia also one type of spores. Juice spore formation. Already we have been discussing from the algal classes how the juice spores are formed inside the juice sporangium. What is the difference between juice spores and aplonospores? Juice spores with flagella like this, we can see in diagram. Flagella present. The flagella may be lateral side like this, lateral side, or the apical side, maybe apical or lateral, and biflagellate. In biflagellate, maybe whiplash type or tinsel type, whiplash or tinsel type, or both types are present. If these flagella absent, that is called as a apnospores. That is called as a apnospores. And conidia formation. It's like a spore formation. Conidia also form, but the conidia formed not inside. Inside the sporangium, outside. The sporangium, conidia formed outside. So this now tell me the conidia formation on the specialized branch that is called conidiophore. They are developed on the conidiophore. We will discuss in penicillium also. Here this is conidia. Conidia. These are this like all shape. These are called conidia. And this is elongated start. This is called conidiophore. And the conidiophore conidia are developed. Right? Chlamydospore, ascospores, iridospores, basidiospores. They are all different type of asexual spores developed inside the sporangium only. Now the another one is sexual reproduction. In sexual reproduction, what is sexual reproduction first?
Fusion of gametes.
Right, fusion of male and female gamete.
Female gamete.
Now how the male gametes and female gamete developed? How the male and female gamete developed? Tell me. In which portion? Sexual reproduction involves the formation and fusion of gametes. Yeah, right, as you said, here gametes. But these gametes developed inside the some of the sex organ. Male one called as antheridium or male gametangium. Female one called as oogonium or female gametangium. Right. So in this different type of sexual reproduction there, that is planogametic copulation. This is simplest type of the sexual reproduction. In this process, fusion of two gametes like this here, two gametes, one plus another gamete. Two gametes opposite sex strain takes place between the one both are fusing gametes and motile flagellated. So here motile flagellate, motile gamete, here also motile gamete. This both motile gamete, both are fused. As a result, formation of
Zygote.
hello, right? That's it. That is planogametic. This is simplest method. And another method is heterogamy or gametangial contact. In planogametic copulation, three types. Two types. One is isogamy, one is heterogamy. What is isogamy? In
The
planogametic,
same.
first of all, planogametic clear. The both gametes are fused. That is planogametic. In planogametic, isogamy and aniso or heterogamy. The simple,
Yes.
simple. In isogamy, both the male and female gametes similar in size. But heterogamy, in general, we can see here. This is isogamy. Both are similar.
Same.
In anisogamy, is the both are similar?
No.
No.
No, this is small, male one small generally, and female one large. So the type of called heterogamy, right? I will tell you later another type of ugamy. Ugama, this is the advanced method. Ugama's method is advanced method, right? Next one, gametangial contact. Gametangial contact. In name itself, gametangial contact. That means in which any gamete formation, name itself given gametangium. Gametangium means male gametangium, female gametangium. Both the gametes opposite side. For example, this is male one act as a male, and this is female. The both gametangia, not gamete. Both gametangia copulation attached. Plus they
Yes.
are from this one mycelium only bulb-like structured form. Later, this is increased in size. This, next step, this cells increased, and finally, the both are attached each other. Later, this between the players dissolve. As a result, both the nuclei exchange. Both the nuclei exchange. And then what happened? The male gametangium, antheridium, transfer the male nucleus or gamete into the female gametangium, oogonia, either through the pore or the point of contact through the fertilization tube, direct by pore, or some of the genera fertilization tube also form. So they enter into the female one through the fertilization tube or the pore. Example, Phytophthora albugopithium. And gametangial copulation. Same here also, in this also, direct gametangial copulation involve the fusion of entire content of the two gametangia. Same. As there is finally zygote formed, example, mucor and rhizopus. Spermatization, another type of method. In spermatization, some fungi produce many minute spore-like single-cell structure called spermatia. This structure transferred through the agency like water and wind. Actually, so far, in first planogametic, only traveled through the water, only cell, gametes. But here, in antheridium or male gametangium, the male gametes such as called spermatia are formed, non-motile gametes are formed. This structure transferred through the agents like water or wind or insect. Other specificity to hyphae. So in female gametangium special structure present that is called hyphae or trichogyne. Now these spermatia connected to the connect to the trichogyne. From trichogyne, finally it is fused with female gamete, which is present inside the female gametangium. Example, Puccinia. And next one, somatogamy. Soma means hello. Soma means vegetative body. That means there is no formation of sexual organ. There is no male gametangium, female gametangium. Direct vegetative thallus mate each other. The type of reproduction called as somatogamy. Example, agaricus. So we can see here. This is gametangial copulation. So this is gamma tangent contact, right? This is gamma tangent copulation, spermatization here, spermatization. So already told you this is male reproductive organ, spermatophore from the spermatospores, spermation. This type of small spermation form. Now this spermation separated from the spermatophore and reach the trichogyne. We can see in diagram trichogyne of the female one. Through trichogyne finally reached into the egg cell into the reproductive female reproductive organ. Now and we can see here the somatogamy. Somatogamy, that is this is vegetative mycelium, this is vegetative, both vegetative mycelium mating each other. So this type is called as somatogamy. Is it clear?
Yes.
Hello? Right.
Yes.
Now next one. Classification, you can read yourself. Classification, mainly classification, Alexomilus classification enough. Right, next one. So this is about algae general character. In algae general character, you should write first of all occurrence, vegetative thallus, cell structure, reproduction. That much enough, okay? Right? If they in general character, no need to write about nutrition. If they separated, write about fungi nutrition, you can write about the fungal nutrition type is saprophyte parasite, again saprophyte obligative parasite, obligative facultative, like that.
Okay, sir.
Right? Is it clear? And also economic importance, I request you all, please read fungi classification and economic importance. These both are general. There is no point to explain. So you can read yourself. We have very less time, right? I think this is the
Yes.
last class. And now the albugo asexual reproduction. Let's see albugo asexual reproduction. Is it clear? First of all, algae clear?
Yes, sir.
Yes.
Right. Now let's see. Uh, let me take PPT slide. Right. Are you able to see albugo, cystopods, positras, cruciferae?
Yes, sir.
Right, this slide. So albugo commonly white rust called as white rust because it is the symptoms appear in white color. It is pathogen name albugo candida is a obligate plant pathogen. That means obligate parasite. Already told you what is obligate parasite. What is obligate parasite?
Permanently depends on other.
What is obligate one? That means only grow as a parasite. Right. Their distribution candida as cosmopolitan distribution are known for many countries. Mainly which attack the cruciferous family. And symptoms you can read. Mycelium structure already we discussed in general character of fungi. Branched aseptate here given uh, just read just brief. Branched mycelium aseptate, xenocytic condition, aseptate with the number of nuclei, that condition called as xenocytic, intercellular hyphae inside the host. So this is the structure of mycelium. And here the very important one is reproduction, asexual reproduction and sexual reproduction. Let's see asexual and so these are the symptoms. You can see white patches. Hello, can you see?
Yes, sir.
So like
Yes, sir.
this is called albugo candida brassicaceae family, mainly mustard. Avalu mainly causes the disease in mustards. Radish, mullangi, they are all like this. They are the symptoms. Okay, okay. Now let's see the asexual reproduction. Simple. First, remember. Let's draw a diagram here. Asexual reproduction takes place by means of conidium or sporangium. Sporangium, sporangium, or juice pores, maybe by juice pores or sporangium. Let's see how they developed. First of all, this is the mycelium. This mycelium present in, this is mycelium here. The mycelium present in inside the host leaf. For example, if the leaf internal structure, mesophyll cells like this, these are mesophyll cell. Okay. Inside mesophyll cell, let's take the color, change the color. Blue color mycelium is present like this. This is the all mycelium. From the mycelium, sporangiophores are developed like this. Sporangiophores, this palisade cylindrical structure. This cylindrical structure called a palisade. This is called uh, sporangiophore. It's called sporangiophore. After developed sporangiophore from the mycelium. First mycelium developed inside the host cell. This is mycelium. This is all mycelium. From the mycelium, sporangiophore develop like this. After that, the tip of the sporangiophore, the cytoplasm of the tip, the like a swollen bulb-like structure develop. This bulb-like structure formation. The next, this first one. The second one formed just below the first one and push the first one to the upright direction. Like this, the number of sporangia developed as a chain. First one, second one, third one, like this number of. But here the final old one is top, young one is base. So this is called as basipetal succession. This type of called as basipetal succession. And after formation, this sporangia, this is called sporangiophore. Remember, this is called sporangium. So anyway, after formation sporangium, each sporangium interconnected with mucilage layer. This is mucilage layer interconnection. This is mucilage layer. So when it is exposed out weather, this mucilage layer dry up, evaporate. As a result, the sporangia separated and take sufficient place. If the temperature below 10 degrees centigrade and water availability present, in that situation, actually, this sporangia, from the sporangium, juice pores are developed. Juice pores are developed. Now the juice pores developed into, that means attack into another host leaf and developed into, for example, these juice pores, when you attack the another host leaf, first of all, they set up these two flagella and form the germ tube. This germ tube finally developed into mycelium. After mycelium formed, what happened again? This is mycelium formation. Okay. After mycelium developed. From the mycelium, what is developed? Hello.
Hyphae.
From mycelium, what is developed here? This is mycelium. From mycelium, which part developed?
Hyphae.
Hyphae, the part of mycelium. Okay, hyphae, the group of hyphae called as mycelium. That is only vegetative. After vegetative structure developed later on the vegetative body, which are developed?
Sporangium.
Right, that's it. Sporangium, like this. Here the sporangiums are developed. From the sporangiophore first, sporangiophore, specialized branch. These sporangiophores are unbranched, cylindrical. On the sporangiophore, which are developed?
Sporangium.
Sporangium, right. This sporangium can directly produce again mycelium or indirectly by juice pores. This is two methods. Direct method. In direct method, the sporangium directly developed mycelium. Indirect method, the sporangium later transfer into the juice pores after production of juice pores. Now the juice pore developed as a mycelium. Right? This is asexual reproduction. Is it clear? Hello?
Yes, sir.
Now the sexual reproduction. Let's see the sexual reproduction. What is sexual reproduction first?
Fusion of gametes.
Fusion of gametes, right? So sexual reproduction is oogamous method. What is oogamous? You know isogamy, anisogamy. Oogamous means actually the female gamete not released out, situated inside the oogamous. Only male gamete developed from the antheridia or male gametangia, which are fused with egg or female gamete inside the oogonium. That's it. If you see the diagram here, so here this is copulation method, gametangial copulation. This type of method called as gametangial copulation. Here the female branch. Let me take the pen. So the female branch, the male branch, the female branch later developed into young oogonium. Here the young antheridium male one. What happened later? The oogonium connected, sorry, the antheridium connected to the oogonium. Next, from the antheridium, one of the tube established between the oogonium and antheridium. This tube called as fertilization tube. After fertilization tube formed, what happened? From the antheridium, male nuclei transferred into the oogonium, where the male nuclei, female nuclei, both are fused. Both are fused. As a result, formation, zygospore or zygote, zygospore or zygote formed. After zygote formed, this zygote undergoes to meiotic division and again form as a flagellated zoospores. Now these flagellated zoospores again germinate into germ tube. From the germ tube, which are developed from the germ tube. Actually, this juice flagella set the flagellates and formed as a cyst. From the cyst, germ tube developed. From germ tube, what is developed from germ tube? Hello. So this is germinated as a
Mycelium.
mycelium. From the mycelium, what are developed? Developed
Sporangiophore
organ.
and sporang.
Right. In asexual reproduction, only sporangiophore developed, sporangium developed. In sexual reproduction, in the same mycelium, oogonium and antheridium are developed. That's it. This is called life cycle. Is it clear? Any
Yes.
doubt in this? Okay.
Yes, sir.
So this is life cycle, right? Life cycle. Di- Diagrammatic graphics. This is only the letters. In above side, asexual reproduction. Below side, sexual reproduction. Remember, albugo, asexual reproduction by conidia or sporangium. Actually, these are sporangium is better word. Sporangium. From the sporangium, juice pores are developed. You can see another line here. In direct method, again this conidia or sporangium germinated into albugomycelium. Here albugomycelium, write down mycelium. Albugomycelium. Right? In indirect method, zoospores are formed, zoospore germinated albugo. In sexual reproduction, oogonium, antheridium, male nuclei, fusion with female nuclei, fertilization, oospore or zygote. It is called zygote or oospore. Meiosis, again zoospore, germination, again mycelium. Simple. You can see in diagram also. Take the picture. Is it clear? Life cycle important. In examination point of view, if they ask about life cycle, you can write the all point and also you draw this life cycle also. You have to draw this life cycle too. Right?
Okay.
Okay,
That
sir.
clear?
Yes, sir.
Based on this, you can read penicillium also. The same, if take the penicillium from the mycelium first, if you take asexual reproduction, in asexual reproduction takes place by conidium. Conidia developed on conidiophore. Conidiophore developed from the mycelium. That's it. Simple. Paxini already you must have learnt in practical training classes, right? You use Iridium use for, Teal use for. They're completed or not in training classes.
Yes,
Hello?
sir.
Right.
Yes, sir.
So,
Ah. Anything else? Draw this life cycle.
Okay, sir.
Take a picture. It's better to take picture. Right. So if you take penicillium also, the same similar to the albuco, asexual reproduction. And the same somewhat similar in sexual reproduction also, penicillium. Right?
Okay.
When I quit the class, any doubts?
No.
Right. Thank you all and all the best.
Thank you, sir.